Great. Welcome, everyone. Let's maybe get started as people are still trickling in. It's great to have Professor Chelsea Rochman today to finish off our week with presentations of the World Water Day celebrations at the University of Birmingham. Chelsea is assistant professor at the University of Toronto and is a co-founder of University of Toronto TRASH team. But she's also a scientific advisor to the Ocean Conservancy and explorer for National Geographic. Her team is focusing on freshwater and marine ecology, ecotoxicology, environmental chemistry, and conservation, and in particular focusing on the impact of anthropogenic debris, plastic in the environment. And Chelsea gained a PhD from the University San Diego State and UC Davis, and then followed on with a postdoc at UC Davis before starting to work at the University of Toronto. And I hardly have to introduce her to you, probably. Many of you will know her from her publications throughout the highest impact journals, from Science to Nature, PNAS, on plastic, really from very small local scales up to global scale plastic pollution issues. And Chelsea is talking today to us about microplastics in our watersheds, their sources, effects, and local solutions. And Chelsea, it's fantastic having you. Really looking forward to your question. And if people have questions, sorry, to your presentation, if people have questions, please put your questions in the Q&A box. You can do that during Chelsea's talk already or afterwards, and Chelsea will be around after her talk to answer your questions as well. Over to you, Chelsea. We're really looking forward to your presentation. Thanks for being here.
Wonderful. Thank you so much. Can you hear me okay?
Yeah, very well.
Okay, perfect. All right. Well, thank you, Stefan, for that introduction. And thanks so much for inviting me to come and give a presentation and to celebrate World Water Day. Because it is a celebration of World Water Day, what I wanted to do is start with just sort of a bit of a background on the issue, talk a bit about why it matters, a little bit about what we know about the science, and then spend some time talking about solutions, and specifically mostly to microplastics, but I'll talk about the issue of plastic pollution in general as well. So I'll start out with a bit about the story of plastic pollution and microplastic. And what I've been noticing lately is that microplastic pollution tends to be in the news almost every day, but certainly weekly. So for fun, last night I went and posted some of the newest articles from this week that popped up on my Google search, which of course included something here at home in the Great Lakes, but also a recent scientific article that came out about microplastics and antibiotic resistance. And so we're starting to see a lot more attention given to this issue, whereas it used to be that people weren't quite aware that microplastic was an emerging contaminant. So I kind of want to dive into the past a little bit and tell you the story of how we got here to this day where we talk about this issue all the time, before I go into detail about microplastic as a contaminant. So over the last 5 decades, actually, the evidence about plastic pollution in our environment has been building, with a slow start followed by a really busy decade. So I'm going to highlight one thing from each decade that I think were fundamental in helping initiate this field. And it actually begins in the 1970s, which is 50 years ago, which is actually quite striking when you're forced to say that. It's been 50 years since Ed Carpenter, who is a professor at San Francisco State University, he reported small pieces of plastic in the middle of the Atlantic Ocean. He reported this contamination in both seawater and also in fish, and he published this work in the journal Science. Now, from what I have heard from people, not from him personally, so the grapevine, is that he got quite a bit of pushback from industry when he published this work and decided not to pursue it as a scientific field. He himself is a phycologist and spends a lot of time trying to understand processes with marine algae. And then as early as the 1980s, the Sea Education Association started monitoring plastics in the ocean, and it was a bit of an accident. So their business was taking undergrads out to sea and teaching them about the oceans. They would take them on these big tall ships, and every group of student would have a different project that they focus on. As they began to notice little bits of plastics in their plankton nets, they actually decided that the plastic project should be one of the projects for these groups of students. And by accident or unbeknownst to them, they basically started the first long-term data set on plastic pollution in the middle of the oceans, which has become incredibly foundational to this field. So if you're familiar with the work of Cara Lavender Law, some of her publications highlight this work. And then, of course, came Captain Charles Moore, a sailor and now founder and director of the Algalita Marine Research Foundation. And he is famous for discovering the so-called Great Pacific Garbage Patch, which has been described as an island of trash floating in the middle of the North Pacific Ocean. Some people say it's twice the size of Texas. Sometimes you hear it twice the size of France. The reality is it's not an island of big plastic mound in the middle of the ocean. It's a bit more of a soup. And in fact, the picture behind the timeline is taken from the middle of the garbage patch, from the bow of a ship. And you can see that the majority of those are tiny little small pieces. So it's not quite how he described it. However, I really think that it's this story that initiated the world to react and some of the work that's been done in the middle of the oceans. But of course, we also have to remember Richard Thompson, who some of us might think of as the godfather of the scientific field, especially those of us that got into this field earlier and really started by reading his work and the work of his students. So Richard Thompson coined the term microplastic in 2004, and this was to refer to those small pieces that were quantified by the SEA Association and also Ed Carpenter and others. And this really sparked a field. You know, he was calling for more scientific research. And I'd say by the decade between 2000 and 2010, you started to see this increase, but you really see a lot of activity in the last decade, for which I guess we're no longer in. And I'm sure we'll have just as much activity, if not more, in this decade. So here we had this growing field that became, I guess, an actual scientific discipline full of people, full of resources in order to answer and ask questions. And now we're starting to see it leading to policy change, which is great. So we've learned a lot since then. And one thing I'd say we learned is that plastic pollution is global. It's not just about the plastic floating around in the middle of the garbage patches. We've learned that, yes, they do accumulate in these subtropical gyres, but they're also incredibly common on our coasts, in urban bays, in our freshwater ecosystems, and even in our house dust. And while most of this material, in the ocean particularly, by weight is large plastic debris, including fishing gear and consumer products, by count, most of them are microplastics. And so I want to focus mostly the rest of my talk on the small stuff, which has really become what I focus on the most in my career. And also, as many of you know, a really ubiquitous contaminant, sort of akin to some of the really persistent organic pollutants like PFAS or PCBs. But of course, they're a bit different than PFAS and PCBs in that they're a multidimensional contaminant. So I tend to think of microplastics as a diverse contaminant suite, the way we think of pesticides as a diverse contaminant suite. There's many different kinds. But the multidimensionality of plastic, I say, because it is made up of many different types of polymers with different types of additive chemistries. They also are broken down from many different plastic products. And they're a particle, they're not a dissolved chemical. And they range in size from the nano range to the millimeter range. They come in many different shapes, and they tend to accumulate chemicals in the environment. So when an organism is exposed to it, it's not just exposed to one chemical, it's exposed to sort of this mixture of a particle, the chemistry in it. And then of course, the size and shape may determine its effect and also tells you something about its source. And what we've really learned over time about this diverse contaminant, as I said earlier, is that it's everywhere. So this includes freshwater and terrestrial ecosystems, and they're similar to persistent organic pollutants in that they are persistent, but as they break down, they multiply and you have more and more smaller particles. They're quite ubiquitous and they can be toxic to wildlife. Something that could be different from persistent organic pollutants is how they move up the food chain and ways in which they transport around the world. And there's still a lot to understand there. But one of the main points I want you to take home from these two figures, or just in general about this ubiquity, is that the scale at which plastic is entering the environment and its decades-long persistence. Plastics are entering the environment at an astounding rate, which I'll show you the number later, and it's become a really important part of the physical environment in ecosystems worldwide. So there's papers talking about how it cycles in the water cycle, the global dust cycle, and potentially even the carbon cycle, or I guess arguably even the carbon cycle. And with this widespread contamination comes exposure in wildlife and then the potential for toxicity. And sometimes we see quite high concentrations in organisms. So sometimes we find hundreds to even thousands of particles in an individual animal. So this raises a lot of questions about the fate in the food web and also the effects. So I want to spend some time talking about that before I talk a little bit about some of the science that informs what we might do about this issue. So one of the things that really, I guess, interests me a lot in my career, and I don't know that I'd say it keeps me up at night, but it's one of the questions I think about often, is what is the fate of microplastic in a food web? This may be where it diverges from your typical persistent organic pollutant. We know that animals ingest plastics. We know that they can inhale them through their gills. We know that they can stick to their skin. Once they get inside the body of an organism, we know that they can transfer. So if they're in the gut, we know that an organism, a predator eats a prey item, that can transfer to the gut of the predator. And we also know that through the gut, you can have excretion. But one of the famous things about persistent organic pollutants is that they're bioaccumulative, right? They're persistent, bioaccumulative, and toxic. And we don't really yet know whether microplastics are bioaccumulative. There's been some evidence starting with Mark Brown's work in 2008 showing that small enough particles can translocate into the cells of muscles. And he found that they can stay there for quite a long time. I think it was about 40 days. So is this evidence for bioaccumulation? Maybe. I think we need a little bit more work looking at this. And then the next question will be, if they bioaccumulate, so if you have this translocation out of the gut into the tissue and it lasts quite a bit, does it magnify up the food chain? And if it does, this means that every particle has to translocate again and again, which could lead to hypotheses about trophic dilution or growth dilution. So because of the fact that it's translocating a particle is difficult, although excreting a translocated particle might be more difficult. So these are questions I think about a lot, which will likely impact the effects. But regardless, as we try to understand this process, we also are starting to understand quite a bit about the effects of microplastics in ambient environment. So I wouldn't say we have all the answers today, but I'd say that we've learned a lot and we have enough answers to start to think about the science we need to do to inform risk and also start to use the science we have to inform risk. So I'll spend some time talking about the different ways in which microplastics can impact an organism. So first, I'll just kind of go over the modes of impact. So one mode of impact would be a physical effect. So this is like actual from the particle. So here on the left is a study from Dang et al., which found that microplastics translocate into the kidney and the liver of mice in this case, with some evidence of inflammation and also changes in gene expression. The translocation of the foreign particle is what's thought to have caused the inflammation and the changes in gene expression within the organism. In our own lab, my graduate student Kennedy Bucci has tried to isolate the chemical effects from the physical effects. And she finds that if she exposes fish in a beaker to the physical particles, or to fish in a beaker where the particles are kind of behind a bag and all that's available is the chemicals from the particles to leach out, that she sees an increase in mortality with the physical particles available to the fish, and of course at a higher concentration in this case, because we know the dose makes the poison. So it seems that a physical effect is certainly possible. Bart Coleman's work shows this as well, and he considers it a food dilution effect. If they're eating plastic and they're not eating their food, then they're suffering from nutritional value, which can of course affect growth, development, and survival. But we also know that there are chemical impacts, and these chemical impacts stem from two different things. One is the plastic itself. Plastic enters the environment, yes, as a particle, but it is also made of different polymers, and they have different chemical additives in them. For example, tire dust is loaded with different types of fillers, and it has metals and it has benzotriazoles. And then the new chemical that came out, there was a paper in the journal Science from Tian et al. 2020, I believe, showing a compound in tires that was toxic to wild salmon. We also know that there's other additives like phthalates and bisphenol A that can have endocrine disrupting effects. So here you're looking at work that also came out of our lab from a postdoc, Anna Kolomaheka, who was working on a project with Environment and Climate Change Canada, looking at the toxicity of the leachate from tires in fathead minnow. And what you see here is the reduction of the pigmentation that you have here in the control within the fathead minnow. We're trying to understand what chemical causes this, and also how does it impact them as they grow up. We know we see increases in mortality, changes in heart rate, we're seeing deformities. The question is, do these last? Does it affect the population, etc.? And then there's also work, here's an example from Kim et al. In 2020 that found that the toxicity that seemed to be observed in C. elegans was driven by the chemicals in the plastic. So that's the additive. Something that's also discussed is the fact that plastics, when they enter the environment, they start to accumulate chemicals from the water around them. And so this is through adsorption and absorption of metals and persistent organic pollutants that are already in the water. Now, there's no doubt that these animals are exposed to these chemicals already. So whether or not plastic adds more of these chemicals is one question, but often the theory, what people predict now is that it's not a huge source of these chemicals. But now you have the bit of this multiple stressor where you have the plastic particle, the additive chemicals, and also this extra sorbed chemicals. And so what we've tried to do in our lab, and this is a study from my PhD, and here's more work from Kennedy, actually the same study, that's looked at both a control, of course, with no plastic, and here's a healthy liver. And here what you're seeing in the control is how many deformities. So you can see the controls are pretty healthy. Fish that are exposed to just plastic taken off the shelf and ground down. So you might see some signs of extra deformities. You might see some signs in this case of basically a lesion in a liver that might be tumor promotion, but we don't know. It's not a full-blown tumor. It's just a focus of cellular alteration. But then we've also taken plastic from the environment and ground it down and exposed animals to it. And here is where we find, in this case, a full-blown tumor in the liver of a fish and more evidence of deformities. So this suggests that as you sort of increase that chemical cocktail, you increase that toxic effect. And so the question is, the fact that they accumulate these chemicals, does it not matter because those chemicals are there already? Or is the plastic adding a multiple stressor, or adding something that makes it a multiple stressor? And over the years, so I'm just showing you some studies that discuss this, but of course there's been lots of researchers that are building a body of work to show how microplastics impact organisms across several levels of biological organization, going from that suborganismal effect, which really tells you something about the mechanism of toxicity, all the way up to a more ecological level of effect, going up into how it impacts the population or the community. All of these are incredibly important. They help us understand how the microplastics may cause an effect and what it might mean in nature. And so over the years, I've kind of gotten in this habit of trying to do systematic reviews to continue to keep track of the literature, to have that weight of evidence to say, what are we seeing across levels of biological organization? And how does it vary with size and shape and polymer type, for example? So I published 2 studies. The newest one is again led by Kennedy in my lab. She's doing quite a bit of work on the effects of microplastics. And then I had published one in 2016 that I had worked on as my postdoc. And here what we've done is basically extracted all of the evidence where they've detected an effect or not detected an effect. We keep track of the dose that they use, the type of plastic, etc. But for this, we plot them on a chart that has the size on the x-axis and the level of biological organization on the y. And you'll see a graph in a minute where the darker the shading means the more evidence of detected effects. And then later I'll show you some trends with dose and polymer type, etc. Right now, I'm involved in kind of a 3rd round of this, and it's to inform a risk assessment in California because they're mandated to come up with a number, a threshold, a risk threshold that basically will trigger a monitoring effort or even source reduction policy. So this work sort of continues, and I'd be happy to answer questions about it after the talk. But I'll start with what we've found in the most recent published meta-analysis and systematic review. And here you're seeing again, where you have shading, it represents the number of effects that were detected for each size of plastic and the level of biological organization. This is macroplastic, so not the focus of my talk exactly, but it's important to note that we have more evidence for demonstrated effects for macroplastic, and particularly at the higher levels of biological organization. That makes sense? We're not dosing animals in a lab with macroplastics very often. Instead, these are observations of animals washed up, entangled, etc. But here you can see that there is evidence of the effects of microplastics across several levels of biological organization, from suborganismal to more ecological, and that it is growing, because if we look at the first one that I did, published in 2016, but literature through 2013, and this is actually just literature through 2017, because you have to stop somewhere. The process for peer review takes a while, but you can see a real increase, not just in the number, notice the scale changes, but also in the levels of biological organization in which we're studying. So this is good. But at this time, the one thing we noticed is that the first time we did this, there were very few studies that had sought to test an effect, but didn't detect one. And one of the reasons is because people were yet to really be doing dose response curves. So there was less studies that had a lower dose that didn't cause an effect. And there were also much fewer studies and people were using higher concentrations. Now we're starting to see studies that use a much more broad stretch of doses. So you would expect for people to be testing hypotheses about effects and not detecting any of those effects. So it makes sense that you now have these non-detects and detects, but you actually have just as many non-detects as detects. And so we were curious, you know, in addition to dose, what might be causing this? And this is important because as we think about building a risk assessment, we have to ask that question of, are we just saying what concentration of microplastics causes an effect? Or do we actually have to take a bit of a step back and say, do we need to take into account the size, the shape, the chemistry that's involved with it? So this is the other question, I guess, that keeps me up at night, which is what makes an effect detected versus not detected? What factors of this multidimensional contaminant are causing effects of microplastic? Is it the dose? Is it the shape? Is it the type? Is it the additive chemistry? And then of course you have these differences in taxa, the size of the organism, but also its physiology and its biology and its ecology. And then of course the experimental design matters. So what I'm going to show you here is just us sort of digging into this with this first set of data, which we're now digging into even further with the larger data set in the study I talked about, trying to answer some questions for the state of California. So here you're going to see on the y-axis the concentration in particles per milliliter. So it's actually just a portion of the data set, not using those that show in mass concentration, because we didn't convert, although we could have. And then whether or not an effect is detected, you'll see the no's on the left and the y's, the yeses on the right. So here you're just looking at a threshold where you can see that at some concentrations an effect is always detected, but at others sometimes you have a yes and sometimes you have a no. And this region is where it's probably driven by some of those other factors. So when we look by shape and polymer type, these graphs are a little bit hard to see what the pattern is because there's quite a bit of yeses and nos. But what this suggests to me is that fibers, which are a more complex shape, may be more likely to cause an effect at a lower concentration than other shapes. And within the California process, what we're seeing is that it does seem like the more so not spherical, so the more complex the shape, so fragments or fibers may have a greater toxicity or be more likely to cause an effect at a lower concentration than others, but there's not a lot of data on this. So I think more work should be done really trying to tease apart these factors. And when it comes to polymer type, there's very few studies that have, or you look at this, you don't see any evidence. And at the time that we published, there were a few studies that had really held everything else constant and tried to look across polymers. Martin Wagner has done some work on this, suggests that there might be a polymer effect, but again, it's preliminary and more work should be done to fully understand it. When it comes to different taxa and different levels of biological organization, this is really more just to show you the diversity that we see, because there's not really a pattern here. However, we do see a pattern with size, in that at those smaller size concentrations, these lighter dots, you tend to have more yeses or all yeses versus no. And when we dig into this a bit further, we also see that size seems to matter. And here you're just looking at acute versus chronic tests. So we're trying to dig into this a little bit more, as I said, trying to understand what factors cause the toxicity. But regardless, if you look at some of the work that's been led by Bart Coleman's group and others, is that if you can collapse all of the factors to try to align the data and you just use a concentration, what he's done is, using species sensitivity distributions, try to understand what concentration of microplastics causes an effect and how many species might that affect. So the HC5 here is the concentration, that hazard concentration that affects 5% of species. And what you're seeing here is the concentrations found in the environment, how frequently, and this HC5 is the orange line. And because you see some concentrations beyond, this suggests that in freshwater ecosystems, which is what this is, there are some places where we're kind of past that tipping point where species are affected. And here in the ocean, similar thing, graphed differently, where you see red, there would actually be some sort of measurable risk. And you see this increasing with time if we continue business as usual. So yes, we have more science to do to really inform our risk assessments, to improve our targets and allow us to be better at predicting and estimating where we have an issue and need to mitigate. But we also have some evidence to suggest that we should be doing this process and that the time to start to act on solutions is now. And there is evidence that concentrations and amounts of plastics will increase. And recently we published a paper where we used a very similar model to what Jenna Jambeck did at her publication in 2015. And actually Jenna was involved in this work to try to both update her number of 8 million metric tons, but also to include freshwater systems, because we think that they're just as important as the ocean. They're not pipelines leading to the ocean, they're also ecosystems. And so we include all freshwater ecosystems, and then we see how, with increasing population, production of plastic, and how waste is managed, what we predict in terms of how plastic emissions into aquatic ecosystems will increase over the years. And you can see with this yellow line, if we continue business as usual, we will increase up to 36 to 90 million tons per year. Today, we're sitting between about 24 and 35. So it's up to threefold if we do not do nothing, but continue what we're doing now. And we really wanted to ask, how much work does it take to get below this threshold of 8 million metric tons? That was this very famous statistic that Jenna and colleagues published, which basically set the world off, saying we need to reduce the amount of plastic emissions. So we don't have a target for how much plastic we want to, we shouldn't put any plastic in the ocean each year, but the reality is, if we set UN goals, there should be a measurable target that we want to reduce to. So we set it at 8 million metric tons until we can do better. And we found that we have a lot of work to do in order to get there. And there's a lot of different solutions that we can do in order to get there. So I'm going to start to talk a bit about solutions. I'll start with a little bit of a focus on big plastic, but then I'm going to flip into microplastic, because I believe that the solutions are a little bit different. We should consider that. So this picture is really just to say there's no one size fits all strategy. There's not a silver bullet solution to plastic pollution, and we shouldn't be paralyzed by that. We should recognize that every little bit counts, and then there's lots of different policy changes we need to tug on at the same time. When it comes to plastic pollution in general, thinking about plastic waste, which is often what's prioritized, I would say in larger policies, but of course for microplastics, things are a bit different. I tend to categorize these into like 3 bins, pun intended. And one is plastic waste reduction. And so that includes banning of single-use plastic items. It includes building a circular economy, because you're reducing plastic waste, you're keeping it in the system. So it includes both of those things. We need to increase waste management. Here, literally, I mean collection from households or collection from industry, and contained. Not allowed to go out into the environment. Of course, a more sustainable waste management system is better than not. And then we need cleanup. And a lot of people say cleanup shouldn't be the solution. It's a Band-Aid. We need to be working upstream. I fully agree. You could very well rank these as first priority, second priority, third priority. But the reality is we need so much work done here, and we continue to see plastics going out into the environment that we absolutely can't ignore this. And I think cleanup is an important part of the solution. And we find that if we really just tug on one of these at a time, the amount of work we need to do is massive. So an example of that is getting up to 85% per capita plastic waste production around the world. That's hard. It's even harder in certain countries. Increasing waste management up to about 99%. In high-income countries, our average is only in the 60%. So I think it's 63. So even that's a big change. And in low-income countries, it can be as low as 6%. And then for plastic pollution cleanup, if we just do that, we have so much to clean. And as I think we all agree, it shouldn't be the first priority. But the reality is we have a ton to do. The other reality is if you spend all your time thinking about microplastic, as I do, these aren't going to solve the issue for some of the most common microplastics we see in the environment. Some of the most common kinds are microfibers and bits of tire dust. And of course, we've heard quite a bit about microbeads, although those are decreasing. Plastic waste isn't really the source of that. This is the wear and tear of products as we're using them. And so here, I want to talk about some of these different pathways for microplastics to get into aquatic ecosystems and talk a bit about what I think are some of the solutions and how I think monitoring, I guess, should be carried out across the world and how that should inform solutions. So some of the pathways that we've looked at closely are agricultural runoff, wastewater treatment plant effluent, and stormwater runoff. Now, there's also quite a bit of work that should be done on industrial effluent. This project that I'm about to share with you did not look at it, but we have done some work on pellets, pre-production plastic pellets from industry. And there's been some studies also on microfibers from textile industry. Another source or pathway, I guess, for microplastics is actually the global dust cycle. Hard to mitigate, so I'm not going to focus on that right now, but that's why you have this picture of dust in the background so we don't forget it. But if you look across the years, more attention is starting to be given to some of these different pathways for microplastics, which helps, I think, inform mitigation. So wastewater treatment plant in orange, you can just see across the years, the number of studies, although the scale, quite low, 0 to 16, have really started to increase. The reality is we focused on wastewater mostly until now. We're slowly starting to see a trickle of studies on agricultural runoff and industrial runoff, and stormwater, I would say, is really starting to take off because some studies suggest that the amount in stormwater might be orders of magnitude more than wastewater. And pesky tire dust seems to come from this particular pathway. So recently, we did a study, which not even recently at this point, this was my postdoc work that I started back in 2014 or 2015 and still haven't published because that's what happens when you get a faculty job and all of a sudden you're teaching and doing all these things, but we're finally getting close. But I want to share the results with you anyways, because it really, for me at least, informs how I think about solutions to the microplastic issue. So what we did was we sampled in 4 regions across North America. So in the California area, so the Sacramento Bay Delta, which is actually just upstream of the San Francisco Bay. We sampled in the Mississippi River, which cuts through North America, huge watershed. Lake Ontario up here at home, so the watershed that feeds into Lake Ontario from Toronto, and then the Chesapeake Bay system, which is also an incredibly large estuary, I think the largest in North America. And in each region, we only sampled, 3 sites where you had either agricultural runoff, wastewater treatment plant runoff, or stormwater runoff. And then we also sampled 3 sites downstream of it, whether it was in the bays or in the lake. And in each site, we sampled 3 samples. So we don't have huge sample size at each site because instead we're in 4 geographies. But our real question was to understand, do we see similar patterns across regions in terms of the quantity of microplastics coming from these pathways, and also the community of microplastics. So do they look the same? Do they have similar morphologies? Do they have similar chemistries? And what can this inform us about sources? So we really wanted to see, are patterns similar, or do they vary locally, which would suggest, if locally, a more local management structure. So we sampled bulk water samples, and we found that microplastics and other anthropogenic particles, so I will note that we corrected for anthropogenic particles, but we didn't take out things like glass beads or cotton textiles. We decided to say microplastics, but also other anthropogenic particles. So what we found basically is that we had these in every single sample. And on the left here, you're seeing the amount in the water body downstream of the pathways. And on the right, you're seeing the concentrations in various pathways. So the first thing I want you to notice is that there's more particles in the pathways than downstream. No surprise, you have dilution in those different water bodies. But the other thing that we thought was interesting is that you have differences in the types of pathways that are larger in the different regions. So in the Sacramento Bay Delta, agriculture seems to be an incredibly important source. And that's the same in the Mississippi River. And here for stormwater, it seems to be kind of the dominating pathway for Lake Ontario, as well as wastewater, whereas wastewater tends to be kind of low or similar in other places. So this suggests that the largest pathway is actually unique across the regions. So in addition to understanding the concentration that matters the most, we wanted to say, how does the assemblage or the community of microplastics vary among these pathways? So for here, we actually treated microplastics in a sample the way we would treat species within a community at a site. And we did this by morphology because we found that there were so many polymer types across the system that there wasn't any pattern. And actually, if we have dug into this a bit more, and it seems like if we truly want to know the diversity of polymer types in a sample, we need to sample more sample, because if we run a rarefaction curve, we're not getting anywhere near where we start to see a flattened curve. So we left polymer type out for now. Instead, we said, by different morphologies, do we have pathways seeming to be similar? So these graphs, what they show you is that the closer the points are together, the more similar the communities are. The further apart they are, the more different they are. This one on the left, this NMDS plot, is across all sites. So you can see wastewater, agriculture, and stormwater, they separate a little, but not in a huge way. So it does suggest unique communities within pathways. And here you can see how they separate by each location. And so here you can kind of see more separation in certain sites and less separation in others, where you can really not tell the difference as well between pathways in the Chesapeake Bay. Now, this is kind of a wonky way to look at this. This is an easier way to look at this. Here you can see the site is each different graph and the proportion of the different morphologies by color. And you can see the different pathways on the x-axis. So here you can see that for agriculture, you actually, in some cases, tend to have this green. This is gypsum. It's glass that's used as a soil amendment. So it is anthropogenic. Whether or not it causes harm, I don't know. And then sometimes you see spheres and you see microfibers, and I think that's from sludge and wastewater. For stormwater, you tend to get this deep red in almost every sample. That's your rubber or your tire wear particles. And wastewater tends to be dominated by microfibers, which is no surprise, and also spheres and kind of like little bits of PVC residue, and probably from the pipes. And so you could suggest that in this case, maybe what you're seeing downstream could inform what you see upstream if you measure your pathways and you measure your water body. Now in the San Francisco Bay, we ended up doing a similar exercise as part of a completely different project. First of all, we found that stormwater had a much higher loading rate to the bay than wastewater, up to 300 times greater than wastewater. And we also found that wastewater was dominated by microfibers and stormwater was dominated by tire dust. So kind of the moral of this story for us was there are many different pathways bringing microplastics into aquatic ecosystems, and we should recognize all of them when we think about solutions to microplastics and also recognize that they're different from some of these pathways we're describing, like the waste stream for plastic pollution writ large. The pathways vary by locations, suggesting that local monitoring is really important. So when we think about how we want to mitigate in Toronto, it might be quite different from how we want to mitigate, for example, in the Chesapeake Bay. And we should all have our monitoring programs like we do for other contaminants, sampling in the pathways, sampling in the environment, and informing mitigation that way. And the different pathways, the fact that they have these different microplastic signatures can actually be used as a tool to help us determine what pathway we want to target. And I'll talk about what I think some of those solutions can be in just a second. And so combined, although it would be really nice to be able to inform across the world where you put your dollars and cents if you're a government, local mitigation seems to be really important. Of course, all of these strategies are probably important everywhere, but if you only have some money and some resources and some time, you probably want to think about what your greatest sources and pathways are. So this kind of comes to this think globally, act locally when it comes to, I think, waste in general and also microplastics. So a few studies that we've done to kind of see what mitigation strategies may be effective. These are more sort of side projects that we've done in our lab as part of the U of T trash team and also in collaboration with the San Francisco Bay, at least for this study, is trying to understand, first of all, how do bioretention cells work, or rain gardens, for example. So on a storm drain, when the storm water runs through, they sort of trickle through this rain garden before they go out into the environment. And we measured the amount of microplastic in the influent as it entered the rain garden, and then we measured it coming out, which by going underground and taking that sample. And so you can see here the effluent is much lower than the influent. We saw about a 90% reduction in microplastic particles or anthropogenic particles in the effluent from the influent, suggesting that these do work. Whether this is scalable is another question, but they certainly should be considered as part of the solution. The other thing that we've tested is, oh, that's a mistake. This is now supposed to say microfibers and washing machines, but we've looked at filtration on washing machines for reducing the microfibers coming out of the washing machine before it goes to the wastewater treatment plant. And we've tested a few different ones of these aftermarket filters where your washing machine water goes through them before it goes out towards a wastewater treatment plant. We've also tested the Cora ball, which sits in your washing machine and attracts microfibers basically as it spins around. We did an experiment. Basically, we have a washing machine in our lab, and we quantified the amount of microfibers if we wash fleece blankets with no mitigation strategy, the amount of microfibers with a Cora ball, and the amount of microfibers with an aftermarket filter, finding again this aftermarket filter is between 80 and 90% effective at removing particles greater than 150 micron in size. We thought this was pretty exciting. We don't think that this means that everyone should go out. I mean, sure, for now you can go out and buy a filter, but we really think there should be legislation that suggests that washing machine manufacturers put the filter in the washing machine and maybe back in the washing machine, because some people have them, and there's rumor that they used to be even more common here. And I don't know how common they are where you sit. I think they're more common right now in Asia and in Australia. But they're effective. And this was just in our lab. We're now working with a nonprofit, Georgian Bay Forever, in a small town of Parry Sound. We've deployed 100 of these in the home of volunteers. The volunteers make up about 10% of what feeds into a wastewater treatment plant, and we are seeing a reduction of microfibers at the wastewater treatment plant scale. Preliminary work suggests it's significant, but we're still digging into that. So that to me suggests this can be scaled up and will make a difference, even if microfibers, even if their only source is obviously not washing machines, there's also dryers and other things. And then finally, this actually could be microplastic and macroplastic, is trash capture devices. We are a big fan of them for removing microplastic and big plastic from the environment, but also for raising awareness and to collect data on the types of sources entering your local waterways that can inform some of those mitigation strategies. So we're actually holding workshops yesterday and today, trying to increase the amount of people around the world that use these, to increase the volume of plastic that's cleaned up around the world, and also to raise awareness and collect data on sources with both a local focus, but also measuring our global impact. So some of these are going to be better at collecting microplastics than others. For example, we catch quite a bit with the sea bin and then storm drain traps, but they're all very effective at collecting litter. So that's my final slide that I'll leave you with today. I'll leave you with a quote that inspires me, basically from Senator Gaylord Nelson in the US, who was the founder of Earth Day. And also I hope, and I think based on time, I have left time for questions. So thanks for listening, and I'm excited to chat afterwards.